So working with EHR data out of the box is actually a challenge that has been around for a while for life science organizations, and I don't think has been solved yet by life science organizations. And I think part of the reason is because out of the box really can mean so many different things with respect to EHR data. So these are the data that are generated natively each day by interactions between patients and physicians at sites of care. But of course, there's no single way that happens. And so those data look different wherever they're generated, hospital to hospital, clinic to clinic, specialty to specialty. And so you end up with this hugely varying amount of data and data type that can also be messy, difficult to interpret, and it's pretty much impossible to work with those data as they emerge from healthcare systems. On the other hand, when you're trying to work with EHR data in a research context, the normalization or regularization that you would typically do to make those data kind of work together can cause you to lose a lot of the fidelity of the data along the way. That sort of compression into a common standard can lose a lot of the nuance that those data in their native format would permit in terms of the visibility that you get to have. That's where we try to differentiate what we can help with, because we are at the point of care, at that point where physicians are documenting in their own language what's going on with patients. We can provide greater visibility into what that intent was and how it remained with the patient in a way that I think makes some of the insights we can derive a little bit richer or different than what you typically get by looking just at EHR data in traditional ways. What's the biggest disconnect between healthcare data and life sciences research? I feel like I could go on about this question for hours, because on the surface it seems obvious, right? Like you take real world data, which are the data generated in real world healthcare interactions, and you should be able to learn from those data a whole bunch of meaningful things for life science companies, how their medications are used, where there's unmet need, how there may be mismatches between what best practices would suggest and what physicians actually do. But I think the disconnect really arises because life science companies have very high evidentiary standards. Their sort of soul is oriented into clinical trials, which are controlled and have super high and rigorous standards for gathering data. But real world data, the kinds of data that are generated in EHRs and also in claim sources, they just don't look like that. And some of that is just because those data are messy, but some of it is also because the kinds of information that we would love from the research perspective just aren't generated in everyday clinical interactions. One good example of this is disease severity metrics. There are specific scales that physicians can use in all sorts of different diseases to capture and rigorously measure how severely a patient is experiencing the condition. But it might take 15 minutes of questions to get that information. The doctor may not want to do that. The patient may not want to sit through it. Now, life science companies oftentimes will say, we want that to get the clearest picture of what's going on. And of course, that makes sense. But that doesn't mean that data sets that don't have those kind of measures or other areas of richness aren't useful for insights. And I think figuring out where the overlap is between what the data can reliably tell us and what's actually of interest to life science companies, that space is what we're always trying to expand and enrich for these clients. When life sciences teams rely on incomplete data, I think the biggest problem is that you don't know what you don't know, right? If you take a look at a data set and you say there's 1 million people in this data set and there's one person in the data set who has my target condition, and therefore I'm going to conclude that the rate of occurrence of my condition is 1 in 1 million, right? That's a really bad conclusion to draw if that data set is incomplete, right? If there's 10 undiagnosed patients hiding in that data set who have the condition, but because of whatever reasons have not yet been diagnosed with it, you just underestimated the prevalence of your disease by an order of magnitude, which is a huge deal. And so relying on data sets that we have reason to believe are incomplete for whatever reason that may be, at a minimum, I think the conclusions you can draw have to be taken with a grain of salt. And a lot of times the way I think folks try to deal with this issue is first go into limitations with their eyes open. It doesn't mean you can only use perfect data sets. You just have to put asterisks where appropriate on conclusions you draw from incomplete data sets. The second thing you can do is try to get more data, because sometimes, not always, but sometimes multiple data sources can help cancel out the deficiencies of each one. But finally, if we can change the incompleteness or the lack of some quality in these data sets to begin with, that can really help, because then we know we're at least kind of drawing conclusions based on the best available information. That's again where I think IMO Health can help a lot, because we specialize in saying whatever's known at that point of care, at that moment between the physician and their patient, whether that's their symptomology, their history, their diagnoses. Let's try to hold on to that as much as we can as those data move downstream. When you do that, you at least have a better confidence that what conclusions you're drawing are sort of the ones most reflected by what was created from that clinical knowledge to begin with. It's not perfect, but it can go a long way to help guard against drawing some of these spurious conclusions.